Welcome to our speaker series, The Use of Mass Spectrometry to Aid ADC Development. Our speaker is Chris Nortcliffe, Mass Spectrometry Manager based at our Specialist Bioconjugation facility in Deeside, Wales, UK. With that, I'll hand it over to you, Chris.
Thank you for that introduction, Emily. Today, I'd like to talk to everyone about the use of mass spectrometry to aid in the development of ADCs, and how here at Stirling we use MS in junction with other traditional techniques to analyze ADCs and other biologics to get the real best quality data on these kind of molecules. First off, I'd like to talk about the biologics field in general and where that's headed in the market right now. And we're seeing a real growth in the field, going from traditional MAB IgG formats into really new architectures. Now, some of these are head towards much smaller features, such as just variable domains, things like VNARs, fab fragments, even down to peptides and cyclic peptides, just taking the very small variable binding regions. And on the other side, we're going towards larger, more heterogeneous structures, such as bispecifics, tetrabodies, other such formats that have different protein chains, that have different challenges for the analytics and how we analyze these molecules. And on top of all these molecules, we've got the addition of drug conjugates of various kinds coming through that add an extra level of complexity in these molecules. So analyzing these can present different challenges going forward. Where we fit in with this at Stirling is our site in Deeside. We're a CDMO and we specialize in the development and analytics of these protein drug conjugates. We have a lot of expertise in a variety of linkers and toxins. So looking at a variety of protein scaffolds, including MABs and other IgGs, up to the small species I talked about, like minibodies and nanobodies, down to peptides and things like that. With a variety of linkers, including cysteine and lysine links, to a lot of toxins that are seen coming through the clinic these days, such as exatecans, PBDs, and those traditional things like orostatins. So we have a wealth of expertise analyzing all these kinds of molecules and conjugating them going forward. The particular challenge of ADCs I'd like to talk about today is going through the partial reduction approach for making ADCs. So with partial reduction approach, we take our IgG of interest and we do a reduction to break apart disulfide bonds into their free cysteines, and then we conjugate our payload molecule to these. This approach is quite well established in the field and produces a very reliable, robust drug-antibody ratio in even numbers such as 0, 2, 4, 6, and 8. Because of the breaking apart of the disulfide chains, when we conjugate these drugs on, we break apart the covalent interaction of the molecule, and the molecules are held together by non-covalent interactions. There can be a couple of challenges in working out exactly what your DAR ratio is for these molecules. One technique for the analysis of ADCs is hydrophobic interaction chromatography, or HIC. Now, HIC is a non-deviation technique that relies upon the interaction of our ADCs with the stationary phase based upon their hydrophobicity. On the left-hand side, we can see a HIC trace with separation of 0, 2, 4, 6, and 8 drug conjugations, and we can take the peak area under these peaks to get a DAR ratio. Now, HIC relies on the absorbance of molecules via UV, so we're seeing an inferring of what each peak is rather than getting a direct measurement. So because we're relying on inferring of peaks, some molecules might not separate very clearly, or there might be overlapping peaks. It can be difficult for some molecules to get a DAR from this technique. Another method we use a lot here at Deeside is reverse phase analysis, often using a PLRP column. Now again, we're doing UV analysis of these molecules, but now we've separated molecules into their heavy and light chains, and we see separation based on their light chains and heavy chains in each conjugation in turn. So here we have a trastuzumab conjugate, and we can see the light chain and heavy chain separating out from the other species. Now this technique can be quantitative for determining a DAR of each chain and combine them together. However, it also relies on inferring of peaks, so it can struggle if things overlap or if there isn't clear definition. If there are any unexpected peaks that can appear, you can be sure what they are. And if for some peaks, such as the H3 peak down here, it can be difficult to know where a peak begins and ends. Now, how can mass spec fit in with these techniques? So MS is a very well established technique in the pharma and biopharma industry for looking at both purity and characterization of small and large molecules. In contrast to the optical techniques I've talked about previously, it provides specific information on the ID of species via their mass to charge ratio. So we can use this to get a lot of information on the molecules we're looking at. On the top here, I've got a light chain and a heavy chain of a protein. We can work out things like glycan modifications on the heavy chain. We can ensure we have any conjugations that affect on there. On the peptide level on the bottom, we can do digestions to work out the particular location of every amino acid in a peptide. And therefore, we can work out exactly where a conjugation has occurred or any of the modifications such as oxidation or deamidation. Now, the mass spec we have here at Deeside is a SCIEX X500B, and we use this at line in the lab on a variety of our conjugation projects. When analysing ADCs compared to just MABs or other proteins, there can be increased difficulty due to the heterogeneity added to molecules with drug conjugation combined with that inherent in the protein. And a lot of optical techniques can struggle to provide the information that mass spectrometry can provide. One example is lysine conjugation. When you have a lysine conjugation, you get a more stochastic level of binding, therefore you get this Gaussian envelope of conjugates. Then MS can be really useful providing a DAR ratio. Another example is ring opening. A lot of payloads have a maleimide present in them that can be ring open to prevent reverse Michael reactions. And this small mass shift can't really be measured by optical techniques, whereas mass spectrometry can be really useful to work out the level of ring opening on various payloads. So the next part of the presentation, I'd like to look at a case study of various payload molecules combined to a transfusion scaffold and how analysis via mass spectrometry can compare with optical techniques. So we selected 3 drugs with different level of hydrophobicity that have been well established in ADCs. On the bottom here, we've got MAE, which is an auristatin, commonly known as vedotin, which is a reasonably hydrophobic molecule present in a commercially available ADC. Going up, we've got MAF, which is also known as mafodotin, again in a commercial ADC, slightly less hydrophobic, but you can see you've got a similar sort of payload to the MAE. They're both auristatins. And at the top, we've got amanitin. Now, amanitin looks very different in the warhead region, which is more peptidic in nature. So amanitin derives from the death cap mushroom and is currently in development for an ADC, and much more hydrophilic than the other 2 molecules looking at. So these 3 cover quite a wide range of hydrophobicity that is present in ADCs. Now, we analyze these conjugates on a well-established Hick method to work out the DAR ratio. For all the molecules, we took a transhuman scaffold and reduced it before doing the conjugations, so they should all have the same DAR ratio as they came from the same reduced stock. Look at the yellow trace first for VCMAE. We can see very clear separation between the 2, 4, 6, and 8 peaks on this Hick trace. We can get a very quantitative DAR ratio of this molecule. Going to the purple trace of MAF, we can see the 0, 2, 4, we've got some separation, they're not baseline. When we get into the 6 and the 8, we're getting a lot of smearing, the peaks are overlapping a lot. You really can't get a quantitative DAR ratio for that molecule. And then looking at the pink trace of amanitin, we're getting a lot of smearing and overlap, even on the low DAR species. This Hick method really isn't separating those species out. So overall, for the MAE, we could use this Hick method to get a DAR, but for the other 2 molecules, a Hick would really struggle to get a DAR for these species. Now, we took these 3 molecules that have been run previously on the generic Hick method and ran them on a generic MS method. Before we went on MS, we did a partial digestion with IDAs followed by reduction to make it into light chain, FC, and FD regions. Now, on the traces here, we can see clear separation in mass dimension from the reconstructed spectra for the light chain, the FD, the FC region, and any drug conjugations that are present. And then we have 3 very different hydrophobicities here. They all ionize well enough for us to get a measure of all the drug species present. And we get a DAR molecule for all these species involved. Now, previously, amanitin was very poor on the hydrophobic interaction chromatography. Because it's very peptidic and has a lot of ionizable points in the warhead, it actually works very well in the mass spectrometry. So what can be a problem molecule for Hick can be a really good molecule for MS. but it shows how MS under generic conditions can be really useful for getting DAR ratio in these kind of species. Now here we have a table of a number of different conjugates we've produced on this trastuzumab scaffold with either DAR of 1, 2, 2.8, 3.7, 4.56, and a high conjugation species. Now again, we worked out the DAR via HIC for the MAE species, and because we reduced all these species in the same manner, they should all have the same DAR. Now comparing the LCMS DAR to the HIC DAR, we can see overall across the table a very good agreement with a small margin of error across all these molecules across all levels of conjugation. This shows how a MS method in a generic MS method can be really useful looking at a lot of molecules. One disagreement here in the bottom corner of the Amanitin DAR, where we're getting a DAR 8 for the LCMS compared to DAR 7.3, is actually isn't an analytical challenge. This is due to conjugation. It can be very difficult to get a high number of conjugates on an antibody, but Amanitin conjugates very well. And it shows how with the Amanitin conjugate compared to the ME and MAF, we just get a higher level of conjugation. Now, the other technique I talked about was reverse phase with PLRP. Now, on the data here, we've got a conjugation with SG3249. For the PLRP trace here, we can see the light chain and we can conjugate in the heavy chain, but L1 and H0 are overlapping. And as they're overlapping, it's difficult to work out how much of one species is contributing to this peak. Therefore, we can't get a quantitative DAR for this molecule. In addition, the H3 peak is very low. We can level ambiguity whether H3 is present or not. Now again, going from a generic HIC method to generic MS method on the bottom with the light chain FC and FD region, we can see all the conjugated species very clearly, and we get a DAR for this species that matches other techniques quite well. The last technique I want to talk about is native MS analysis using size exclusion chromatography, or SEC. Now, most mass spec is taking place under reverse phase conditions with denaturing welfare such as acetonitrile and levels of acid. And this unfolds a protein into its denatured state with a lot of surface accessible sites that can charge. So here on the left-hand side, we've got a spectra for a MAB with between 30 and 60 positive charges in an unfolded denatured state. Now, native analysis seeks to introduce molecules into gas phase in a more folded state, keeping these non-covalent interactions bound to each other, keep it in its folded state as it would be in solution. So on the right-hand side, we can see the same protein, but now ionized via native SEC mass spec. We can see a much narrower charted distribution as the molecule is now folded as it would be in solution, and only surface accessible sites can become ionized. So we've got a narrower charted distribution. What this means is that we can get an analysis of molecules in the same way with HIC, molecule being intact, rather than individual chains after reduction in isomers. So here's how the native MS looks for an ADC rather than just for a MAB. So here we can see on the left-hand side the raw charge state distribution of these molecules. Now compared to the general MAB we saw in the previous slides, things look more complicated because now as well as glycans, we also have drug conjugates present on our molecule. And on the right-hand side, we've done the reconstruction molecule and we've taken the glycans off with PNGs-F. So here we can see a very reminiscent spectra of what we'd see via HIC, with the 0, 2, 4, 6, and 8 peaks being very clearly visible. So look, analytically, it seems very similar in the data. Looking at the raw numbers for this, via HIC, we've got a DAR of 3.98, and via mass spec, we've got a DAR of 4.03. So we're getting very good agreement between the two techniques for a reasonably hydrophobic payload. Now, native mass spec can be very useful for things like bispecifics or other molecules to get an intact DAR ratio rather than breaking apart into light and heavy chain. Tying all this together, Sterling has a deep expertise in the conjugation analysis in a variety of next generation ADCs, and we work with a number of commercial partners on proteins and ADCs across various levels of the developmental pipeline, from discovery all the way up to preclinical and beyond. And it's this broad understanding combined with a specialized lab to handle cytotoxic material makes Sterling a proven partner for analysis of these biomolecules. We have a mass spectrometer inside our lab, and we do analysis at line with all our chemists at various levels of the developmental process. We can get really rapid answers using MS or other techniques I mentioned. It's this combined use of mass spectrometry with traditional chromatography techniques that helps bring a fuller understanding to any biopharmaceutical challenge. With that, I'd like to thank everyone for listening to me today. If you have any further questions, please visit our website and get in touch.